applications that we have for filamentous fungi with alternative proteins, but in a variety of other unexpected places. And that's what I mean by beyond here. And I'll show you what I mean by that more during the presentation. So first up, I want to acknowledge my various collaborators right off the bat here. So Professor Reihong Zhang at UC Davis, along with Lin Chow, Professor Zhongli Pan, Hamed El Mashad, all from UC Davis. Really, want to thank them for all their collaboration over the years while we've been working in this area of filamentous fungi for foods. And we continue to have collaborations today. I also want to acknowledge Shuvro Talukdar, who is a master's student at the University of Kentucky that's been working with me and is responsible for some of the graphs and beautiful figures that you're going to see later on in the presentation. And lastly, Optimized Foods, which is a company, a startup company working to produce mycelium-based platforms and new food products, and has been actually licensing some of the technologies that you're going to see during this presentation. And since this is a science-based talk, I also want to direct you to the right side of the screen where a couple recent review papers that I was lead author of these. The first one being Fungi for Future Foods, which I'll pull a lot of figures and ideas from for this presentation. You can find more from that. It's an open access article. And then the bottom one, which is more broad review about cell cultivated food production and processing in food bioengineering. Both of those articles are open access and available to you. So just want to put those up as resources for you. So for my talk today, I'm going to briefly talk about some background and rationale. I think that's going to overlap greatly with the rationale for everybody in this audience's work. But this will specifically be kind of what drives me and the type of research that I do. And then we're going to meet our main characters, the fungi here. I'm going to give you a little background about their growth and life cycle and their history in food products. And then go into my favorite part, which is about cultivating and processing. So I'm not a biologist. I am an engineer that uses biology. And the cultivation and processing portion of this is where we can really change how these organisms are growing. And there's some really fascinating details about fungi, filamentous fungi, that give us some levers to pull to make really interesting products in the end. And that'll springboard me into my two case studies, which I've purposefully left a little mysterious here. And I'll show you what I mean by those. First one is going to be a story based around an unlikely myco-partnership. And the second one, an unexpected myco-opportunity. So some brief background about myself. So I am from the Southeast of the United States, went to undergrad for biosystems engineering at Clemson University, and then did all of my graduate work, including a postdoc out in California at UC Davis. And in 2021, I was very fortunate to be offered the job at University of Kentucky to lead this bioprocessing and fermentation lab. And I'm pretty early in my career, and the research emphasis is really in bioprocessing and fermentation. And I specifically like to work with fungal and algal biotechnology, upgrading of food and agricultural industry byproducts, including doing things with renewable energy, and then also taking in some modeling around technoeconomics and life cycle assessment. And I always like to say, I'm also a real person, and hopefully that comes across in the talk here, but I always like to put a plug there. Real person that likes real things, cooking, traveling. I play classical guitar, and I've got two cats who hopefully are going to behave during the talk here. All right, so here's some details about the background and my rationale for the type of research that I do. And we're all aware that the population is increasing. We need more food. We need more energy. The population is increasing not uniformly across the world. And that's relevant for when we're discussing and thinking about new types of food products to make, in that we need to really have a global outlook on these things. The same things that work or are accepted in North America, for instance, are not the same types of products or innovations that are going to work everywhere else in the world. And we need to have a more global mindset to make sure that we're tackling these problems and making a big impact ultimately across the world. Food production is going to have to increase, and I show this graph in the middle because it includes the portion of food waste that is going to increase along with the food production. And that really puts more and more pressure on us to deal with that waste in increasingly sustainable and circular ways. Energy production needs to come more and more from renewable sources, and I'm going to make a case in a couple slides to you about how energy, food, population growth, water, all of these things are connected in ways that we can't really take one of these issues apart from each other. But like you, I'm just trying to, answer this question of, how we can support the planet and humanity and all the other biodiversity on this planet into the 21st century. So from my specific field and work is in bioprocess engineering. And what we do here is think about taking what I spend a lot of time thinking about is taking our traditional systems and looking at them in different ways, trying to kind of engineer out inefficiencies or places where we're losing energy, losing nutrients in the process. So if you take a traditional field as an example, here that we're loading with nutrients and water and so forth. As we put these on the field and we grow our crops, there is a spatial and temporal loss of those nutrients and water, also gaseous emissions into the atmosphere. And this is pretty uncontrolled system. We're not really certain how much of these end up in the plant, how much of it goes to the groundwater or runs off, in those types of issues. Whereas if you look at a bioprocess that will cooperate with me. Here we go. You might have a process that looks more like this, with water and nutrients coming into a bioreactor that is growing our microorganisms. We have a central, one pipe that comes out of this. We harvest our organisms and products and recycle the water and nutrients that we can. And whatever is left over from that process, we can deal with and quantify it easier as a point source at the end. These types of bioprocesses also allow us more control of the environment, more control of biology, making products that we truly want and less of what we don't want. It also, in the graphic on the right, allows for this concept of circularity to come more naturally into the designs and allowing for direct and efficient coupling of renewable energy, recycling of nutrients, et cetera, through agricultural systems. And more we're seeing these types, the emergence of the circular bioeconomy, not just for alternative food production or things like that, but also into chemicals and fuels, materials. So these are really things that drive me and the types of things I'm going to talk about today. So now we get to meet our main characters of the story here, the fungi. And this slide, I'm going to show you some pictures from one of my favorite classes when I was at UC Davis by Professor Sturgiopoulos. Put these beautiful pictures together of the macroscopic, beautiful diversity of the forms of mushrooms that we see out in nature. Beautiful colors, different shapes and sizes. And on a microscopic level, everything from our single cellular yeast that we use for, beer and wine production, up to the molds that grow in your bathroom or on your bread that you left out too long, including the mycoprotein that is produced by companies like corn. And as you see in these two pictures, they have these two lives on a more macroscopic and a more microscopic level. And there's a lot to learn and to take in on these two scales and really try to harness all the diversity that's out there. I think we're really still just scraping the surface of the total potential of these organisms. So to give you a little background about fungi and how they grow, talk about their morphology here. And this will be important later on to understand the two major growth forms of fungi. And one is as a yeast form, that is a more unicellular type of growth form of fungi. And the other, which is going to be the main topic of my talk here, is a mycelial form, which we call made up of a thing called hyphae. And in the center and bottom pictures there, you can kind of see what mycelium are, and what hyphae are these little finger-like protrusions that grow outwards in a radial fashion. And the reason that they do this in nature is for exploration, invasion of new food sources. So fungi are these great explorers. They move out in their biomass out into the environment, and they also put up spore structures that help to disperse them even farther away from, and they're kind of looking for new places to explore all the time, and they're very good at breaking down complex forms of biomass, pretty much anything that they could possibly come into contact with. Some fungi can switch between the yeast and the mycelial growth forms depending on the conditions. And so this is an example of where the engineers that design a bioreactor, for instance, need to understand what can shift a fungi from a yeast form to a mycelial form and so forth. And, take that into consideration, because these are drastically different ways of growing, and the fungi can sometimes do both of them. And humans have been using fungi in foods for centuries. This is not a new concept in general. 16,000 BC, there's evidence of human consumption of mushrooms. And in the 1600s, soy sauce was being commercially produced in Japan. And post the Industrial Revolution to now, you've seen this huge increase in biological knowledge and biotechnology. Fungi are being used for producing antibiotics and enzymes in industrial fermentations. Nowadays, we're moving into precision fermentation, cellular agriculture, applications for filamentous fungi. Things like 3D printing are coming up, and we're standing at a place of great excitement and development and opportunity. It's an exciting place to be. But why are we interested in fungi for foods? The first reason is because of their diversity. As I talked about, the species diversity. in all these beautiful different colors and shapes and sizes of fungi. There's just a lot of them out there, and we don't know that much about many of them. But also they produce a lot of products in addition to their biomass. You might not be aware that the citric acid that is used as a food ingredient is not produced from lemons and oranges. It's in general produced from very large fermentations of Aspergillus niger. They're also quite versatile in their morphology, as we mentioned with the yeast and mycelial growth forms, and I'll mention some others, important forms later, as well as their life cycle in supporting both asexual and sexual, very distinctive portions of their life cycle. And they're established in the food industry for the production of biomass. Of course, we know about mushrooms, myco-protein, two examples of that, as well as their fermentation products with enzymes and antibiotics, citric acids, et cetera, et cetera. From a nutritional point of view, fungi biomass can have complete amino acid profiles and offer a good source of dietary fiber as virtue of having chitin in their cell walls. They can deliver textures and flavors, be quite delicious, and potentially mimicking muscle fibers because of this way that they grow out in the hyphal form. For the environment, when we grow things like fungi in bioreactors, we have tight control over the way that we use and reuse our resources like water, nutrients, and energy. And of course, there's the ethical reasons for being interested in fungi, especially in the alternative protein space, for instance, with animal versus myco-protein productions. And I don't think I need to describe that in too much detail to the audience here, but it's an important consideration. So here's speaking about the nutritional profiles, compiled some nutritional profiles for the amino acid profiles for the essential and non-essential amino acids of different edible fungi on the left-hand side. The Aspergillus species are ones are historically used in koji production for things like soy sauce in Japan. And Fusarium venenatum is the organism that corn uses for their mycoprotein. And then on the right-hand side, you can see the nutritional profiles from a more macroscopic point of view here with very high producing protein strains of different edible fungi there between 45 and 50% protein. We also have a good source of dietary fiber as well as other macro and micronutrients. And increasingly, as we talk about fungi being applied in future food products, you're going to see more and more talk about circular and precision manufacturing principles coming into play here. And that's kind of shown in this schematic on the right-hand side, taking agricultural and food byproducts by virtue of their cheap cost for one, but also because we want to use these instead of letting them go to waste and just throwing them in a landfill. As I'll show you, they're quite good sources of nutrients for fungi, which are organisms that are used to using byproducts in nature. They're some of the great decomposers of the world. And so using those as a starting place, we can go down our cultivation into our processing and doing some quality control, ultimately making food products. And as I mentioned, one of the things that I want to drive home in the talk today is alternative proteins are one of those food products. And there's a variety of other things that if we let our imaginations go, we'll see that there are other applications out there that can be quite complementary to the alternative protein space. So now to talk about how we cultivate them, process them, and get into some of the research aspects of this. We'll again talk about the fungal morphology. So with this mycelial growth that we have, When we're growing them in submerged fermentations, what you're probably thinking about when you think about a fermentation is being, in liquid underwater, which is not the way that we grow things like mushrooms, which is a solid state fermentation. We can use that media and the way that we load the cells in, the way that we agitate it and so forth to create these things called fungal pellets. And what the fungal pellets are and what they look like is shown on the right-hand side. Fungal pellets are basically little spheres or little balls that form when you're growing fungi in this form. And they're macroscopic. They're quite large. They're millimeters to centimeters in size when you grow them. And, you can make them quite small or quite large depending on your application. And the reason why these fungal pellets are important to talk about from a bioprocess engineering standpoint is because the fungal pellet itself is itself a little microenvironment. And that's what you see in the center picture, which illustrates how as you move from the interior of the pellet out to the exterior, you have a gradient in the concentration of things like oxygen and nutrients. The interior, the very center of that pellet, could be quite oxygen limited of other nutrients in there. Therefore, the cells in there are not experiencing the same thing that the cells on the outside of the pellet are. They might be more stressed out in there, producing different types of products that end up getting into your process. And controlling that and making sure that we are delivering enough oxygen, enough nutrients to the whole pellet that we're interested in working with here for whatever application we have is an important part of the design process. The benefits of using pellets like this as opposed to their more unicellular growth forms or even growing them in great masses, clumps, is that they're very easy to harvest. It's pretty simple for us to take out things that are millimeters and centimeters in size, just coarse filtration with cheesecloth or something like that. Don't have to do fancy centrifugation and things like that. But also we get some benefits in mass and heat transfer and broth viscosity as a result of doing this as well. And when we're growing the fungi, we take a lot of time and consideration to the cultivation media. And like all of the other types of organisms that have been talked about in this seminar series, the media selection and design and optimization is a critical component of fungal cultivation. For fungi, it's really about sugar. Sugar is the expensive part of media. Macro and micronutrients are also required for the cultivation media. And the media exerts great influence over the growth of the microbes as well as the final product that you get. And increasingly, we're using more and more byproducts from the agriculture and food processing industries in the cultivation of filamentous fungi, as you can see in the table on the right-hand side, which has 3 different byproducts being used as the initial substrate for a variety of different fungal species, producing their biomass as well as the protein yields that you can see there. In the bottom, you can see a specific case done by my colleague Lin Chow with Professor Ruihong Zhang at UC Davis, using a very abundant byproduct from the almond industry in California, which is using a specific part of the plant called the hull, which is produced actually in greater quantities than the almond kernel itself in that industry, and essentially shares a lot of characteristics with peaches in that it's high in sugar and has some of the same flavor and antioxidant compounds. You can easily extract that sugar out of these almond hulls, inoculate your fungal spores, and grow up this beautiful fungal biomass with some of the characteristics of that almond hull For instance, its sweetness, earthiness, and flavors, as well as its health benefits from different phenolic compounds, things like that in the final product. And when we're working with different types of media and trying to optimize media, we'll look at certain things to determine how long it takes to grow the biomass, how well the fungi is adapting to that new material. And so we'll get growth curves like you see on the left-hand side, which is utilizing, consuming sugar to produce biomass. And one thing you can notice from that graph is about a 50% conversion efficiency between the sugar and ultimately ending up in the biomass at the end. Of course, we have losses from this. This is a biological system, not perfect, in that we're not just producing biomass here. These are living, breathing organisms that are respiring out carbon dioxide. They're secreting other products of their metabolism. And you can look at the graph on the right to see the individual sugar consumption and see one of the benefits of using fungi, and that you can utilize a great variety of different sugars. Some fungi can use even 5-carbon sugars, which was not the case with the fungi that you see here by the xylose curve there. That didn't really change much, but we didn't have much to begin with in this material. And once you've grown up all of these nice fungi biomass, what do you do with them? And one thing that I'm sure, at least I am interested in, and perhaps some others in the audience are around 3D printing. And so for my own personal research program here, we're developing this bioprinter that you see, or developing projects around this 3D bioprinter that you see in the picture and video here, which is a 3-head pneumatic system that uses needle and nozzle type of extrusion for high precision printing of any structure you want, essentially. And here we're printing out some, you can print out scaffolding material with this. You can load in your cells here, potentially being fungi or other types of microorganisms, and use them in your biolinks as active materials and also as their own scaffolds. And so I don't have a huge amount of data to show you, sadly, on this, because it's like literally a few weeks old in my lab, but I just wanted to show you all because it's so cool. And I think there's going to be a lot of applications for this with our work moving into the future. So that will jump me into my first case study here, taking us away from the fundamentals of filamentous fungi cultivation, all these things, into an unlikely myco-partnership. And the partnership that I'm referring to here is about a relationship between fungi with some other organism. And in this case, that other organism is microalgae in the form that they exist in nature as lichen. And I have, in parentheses there, unlikely. Well, it's not really unlikely, right? Lichen are everywhere in nature. They grow in these crazy, harsh environments. On the backs of rocks and on trees and so forth, hardy organisms. Not really an unlikely or novel topic as far as nature is concerned, but as far as food production is concerned, there's an interesting relationship that we can kind of tap into in the evolutionary history of these two organisms to work with them for food production. And how we do that is by basically using filamentous fungi pellets as a scaffolding, a living scaffolding for the attachment and proliferation of microalgae cells. And you can see how that process works in the diagram here, where you basically just drop in these fungal pellets into a nice, happy algae culture. And then this magical thing happens where these two organisms that, are used to living with each other in nature kind of naturally come together, and the algae is brought to the surface of the pellet in this really interesting fashion, forming this algal and fungal coating on the outside of the pellets. And why are we interested in microalgae for this? So microalgae bring a lot of benefit to alternative protein production in that they are, or you can grow a lot of them autotrophically, phototrophically to be more specific, in that they use inorganic carbon, CO2, as well as light energy to grow up. And as I mentioned before earlier, in growing the filamentous fungi, there's a portion of that sugar that doesn't end up in our fungal biomass, just goes out as CO2, which is, a great food product for these microalgae. And in fact, studies have shown that when you do this co-culturing system with the fungi and the microalgae, you can actually get some synergy depending on if you're using the right combination of fungi and microalgae together, where you end up producing more fungal and algal biomass in the final product of the co-culture than you do with the two cultures independently. Microalgae is cultivated in the lab in something that looks like this. This is in my lab at University of Kentucky with various scales of microalgae production. In the real world, microalgae is made in very large scale outdoor raceway ponds, or also in photobioreactors and these kind of tubular arrays and some other various ways of growing them as well. But we have been interested in using microalgae for the food applications, specifically with this species called Hemetococcus pluvialis, which is on the right-hand side. And in the picture on the top there, you see a very specific portion of the life cycle of microalgae there, where for all the rest of its life cycle, it looks green, like a nice, happy microscopic plant that it is. And then later on, it turns into this beautiful red color from the astaxanthin pigment that it accumulates at the end. And this nice bright red color I will leave to the imaginations of all the food scientists in the room for how that's an important color for alternative protein production. But I'll also say that this is a very valuable antioxidant material with health properties. And that's just one of the things that the algae brings to the system. It can also bring even higher protein contents, different forms of fatty acids, and so forth. So the combination of these two things is a pretty compelling case for alternative proteins. Microscopically, if we look at the filamentous fungi pellets that are participating in this immobilization process, you might see, or this might surprise you, what they look like when we look at them under a scanning electron microscope, in that these pellets that look like very solid spherical things, they just look like solid balls. Macroscopically, when you look at them under the microscope, you see what they really are is a complex, almost sponge-like material. They remind me of those little moss balls that people buy and will have as pets in their aquarium. It's a continuous interweaving of these hyphae into this what looks like a more organized structure than it really is on a macroscope or on a microscopic level. And when you use them as a scaffolding material for microalgae cells, this is what you will see under the microscope. So here, by following the rows, we see the surface of the filamentous pellet. And then we have moving inside the pellet into a cross section about a millimeter inside, and then looking at an entire pellet just directly under the microscope. And what you see before the immobilization process is these little finger-like protrusions. Those are the hyphae of our fungi. But then afterwards, you see everywhere inside of there, these microalgae cells bound to the filamentous hyphae. But they're not just bound to the hyphae, right? They're in clumps, they're bound to each other. And if you look inside a little bit more closely, you can see as evidence of EPS, extrapolymeric substances that look like little spider webs that help to bind the algae together, actually, and then come into the little fingers for the fungi to grab. And this does not just happen on the outside of the pellets like we might imagine. It goes inside of the pellet, and a millimeter inside of the pellet, you can see a lot of evidence of cells migrating into and being immobilized inside of there as well. So this is a truly three-dimensional and living scaffolding material that is edible, is safe, all of these things. We can control the geometry and the texture, structure, and so forth. Under a light microscope, I just think it's interesting, the picture at the very bottom there. You don't have to do a lot of this fancy electron microscopy to really see straight through a fungal pellet with your light microscope. And in the bottom right picture there, you can actually see the microalgae cells that are immobilized in that scaffolding just with a simple light microscope technique. So of course, when we're doing this, we are interested in being able to control things. And specifically here, talking about controlling the algae content of the final product, being those pellets that we're getting in the end. And it's sort of serving dual purposes for us. The microalgae are historically hard to get out of solution. They're super tiny and we have to centrifuge them and all these energy intensive things. But here we can bring them to the fungal surface. We can change the content of that resulting pellet to, depending on the loading of fungi that we're bringing into that system. And in the graph on the right, you can see that actually you can get more than the fungi's initial weight in pellet of algae attached and algae content in that pellet than when you started. And that's really cool. So we have quite a large range that we can change depending on the attributes of the product that we're looking for, whether we want more or less of that pigment that I was talking about, or more or less of a grassy or marine type of flavor that comes from the microalgae. Fungi clearly don't deliver as much. And this works with other cell types, not just microalgae. Here we have an example of animal cells with third kingdom caviar from Optimized Foods. And they use a fungal scaffolding there to attach animal cells from sturgeon to make caviar without growing that in a fish and harvesting it from the fish. On the right-hand side, you see this being applied to yeast cells that are immobilized and have in that fungal mycelium, have applications in beverage fermentations. And I'll leave you to reference my colleague, Dr. Marino Garcia there with his work on that application. And so that'll lead me into my second case study about the unexpected myco-opportunity here. And this is the part where I want to kind of bring home the idea that You probably expected me to talk a lot about alternative proteins in this talk, right? There's a lot of opportunities in other areas of the food portion. So we're really expecting a lot of alternative protein talk, but were you expecting me to then talk about boba and milk tea, textured beverages? Um, Probably not. But one of the things in working with these nice spherical pellets that we get from these fungi was the more we worked with it, the more we looked at it, we're like, wow, that looks a lot like the boba and these tapioca pearls that we all enjoy from the boba shop, right? And so then we started coloring them, adding flavors to them. You see lots of examples of using natural flavors and layering these things so that we can get, different flavors and colors on the interior versus the exterior, creating this really interesting transparent kind of covering over a dark center. The pellets, which you see on the far right side, we call those the boba eggs. Um, and then also doing things like bringing other biopolymers into the mix to deliver different textural experiences to the consumer. So this is examples of complexed with agar, alginate, and gelatin pellets. And that's because we're looking for a different texture with a textured beverage application of these fungal pellets than we are with an alternative protein application, right? And so we can change the chewiness and the bounciness by virtue of this complexing process. And this is what that looks like in process form to create multiple layers of different colors and flavors, textures with a process that's currently a patent pending process at UC Davis and a letter agreement with Optimized Foods out there. To make a product, we call it myco-boba. It's had a lot of names and it'll probably have more in the future, but a process that creates these nice fungal balls that can add a lot in a healthy and more sustainable way to these types of beverages. Another thing is you notice the bottle there. Another interesting thing that we noticed when working with these was that they were quite a bit more shelf stable than traditional tapioca pearls are as well. And, by virtue of these are fungi, chitinous cell walls, hard to break down, these types of things. So one thing that this opened up the opportunity for was into a ready to drink and RTD market application for using filamentous fungi to make this myco-boba, put it into a can or a bottle and put it on the shelf and not have it turn into that a kind of boba goo stuff that happens several hours after you get it from the boba shop traditionally. And we looked at that into the texture stability of the uncoated pellets as well as the ones that we coated with these various biopolymers, and looked at from a texture profile analysis standpoint how their chewiness and hardness, cohesiveness, adhesiveness were changing during storage for 1 month at 4 degrees Celsius. And you see the various performance for the different types of polymer coatings and kind of directing us towards different coatings for different applications. And so the last thing that I have to talk about, talking about this, is to show you some nutrition facts about this particular product. And as you might expect, it's lower calorie. It's got less starch than a tapioca pearl has, and it has a lot more fiber and protein as well. And so it's a healthier kind of alternative. And so we're looking forward to seeing if we can kind of bring this thing to market. And so with that, I'll kind of close out the technical portion of what I wanted to talk to you about. And hopefully that was, brought you into some new ideas in the fungi space for food. And before I completely leave you, I just want to give a short plug because of the nature of the audience here to a special issue of the Journal of Visual Experiments, which I'm serving as a guest editor for. This is a really cool journal that produces these video types of articles for methods. And we're doing a collection, which I am a guest editor of, for the production, characterization, and evaluation of cell cultivated foods. And everybody that's on the call here, if you're working in this space and have some methods that you'd like to get out there and share with the world in a really progressive, interesting new type of article, please contact me. I'll be happy to bring you in to the fold on that. And lastly, it's the last plug I'll give. Just want to really thank you for your time. It's been a true pleasure and an honor to be able to give this and to be invited to give this talk. And I'm really looking forward to fielding the questions and continuing the conversation with you. So thank you so much.
Thank you so much, Tyler, for a phenomenal presentation. The chat was just blowing up throughout. Just marveling at microorganisms. I think whose beauty was on full display in your presentation, but also just thanking you for putting together such a detailed, comprehensive, informative presentation, and for sharing your research in an open access manner, because that does so much to advance the state of scientific knowledge in the world. So many thanks to you. Lots of joy and gratitude directed your way from our community.
Thank you.
Yeah, if you'd like to, you can stop sharing your screen at this point and we can move into our Q&A portion. Those of you who are there in the audience and haven't yet asked your questions to Tyler, please do feel free to drop in your questions into the Q&A bar. We have 33 questions and more than likely we'll not get to them all. But as Tyler has very generously offered up his contact info to continue the conversation if there are any questions that we don't get to today. All righty, Tyler, do you feel ready to dive in?
Oh yeah, let's do it.
Awesome. The very first question here, there are lots of questions around the kind of nutritional composition of filamentous fungi. So maybe we can start there. The amino acid and nutritional profile that you showed was super interesting, but this attendee is wondering whether the profile is very much influenced by the substrate that the fungi grows on, and if so, kind of what considerations should we have in mind for that?
Oh yeah, I love that question because it comes back to the, a broader point that I like to make about just growing microbes in general, and the power that engineers and us working in the bioprocess engineering space, we get to have a lot of say over what these organisms end up looking like compositionally in the end. So we can't change to begin with, there are strains that naturally will have various profiles. They have their means of expressing their various pathways to produce, for instance, various amino acid profiles or lipid profiles and so forth. So we can change that. In my lab, we don't often change that as much as some others. Genetically, we tend to work with those that are already out there more. So the other thing that we have at our disposal in the bioprocess sense is by what we expose these organisms to, and that changes how they grow. It changes what the composition of their profiles are in any of these individual components. And if you, for instance, it's been shown that if you stress them out, for instance, with a nitrogen type of limitation, you can greatly change and modulate the amount of lipids that are produced. So you can go from trying to produce a more protein-rich type of final product there to a much more oleaginous product just by how you're exposing it to its environment. And that goes from not only the things that we can load different chemicals in there, peroxide and salts and things to kind of stress them out, make them change their metabolism that way. But also, as I said, when working with fungi, it's trickier than even the unicellular organisms that we work with, for instance, with the microalgae, in that even within those little pellets that we have, those cells are experiencing their environment quite differently. And, from a bioprocessing engineer's perspective, we're looking at how big are those pellets? How big is the profile of concentration of oxygen and nutrients throughout that profile? And that can change our final biomass as well. So, those are all things I think we can talk probably a full other hour about how we can change that composition, but it's a really important aspect of doing this type of work.
Thank you so much for that super detailed response. Staying on this theme of kind of the nutritional world and allergenicity, well, there's one question that's just a clarifying question on whether the nutritional panels you showed were for the mycelium or fruiting bodies of the various edible fungi. And then more broadly, asking for your commentary on using mycelium versus the fruiting bodies for various alternative protein products.
Mm-hmm. Yeah, that's a great, and I couldn't even touch on all of that, the details. It's another thing about fungi, and that, when we start talking about fruiting bodies and we start talking more about mushrooms, well, mushrooms are a very particular part of fungi, and most of the ones that I was talking about today actually belong to an entirely different class of fungi, more similar to mold type of fungi, find on your bread and that type of stuff, that don't have that part of their life cycle to really make distinctive fruiting bodies like that. And so all of those figures that I was showing in the presentation were for the mycelium biomass itself, usually in the form of the pellets that we get. And of course, when you're working with those species that can form mushrooms, we have a whole other processing component to handle from their life cycle standpoint, and kind of inducing them to make a fruiting body, which is actually a distinctive part of their life cycle and creates so many different types of, the composition of that portion of the biomass can be quite different from the mycelial roots, if you think about them that way.
Mm-hmm. Yeah, that's really fascinating. There's you made a good connection or a good lead into this next question about food allergenicity. And this attendee is wondering whether allergies are a concern of fungi-based food, because mold is a common environmental allergen and maybe causing some cross-reactivity. So just curious for your take on whether there's a cautionary tale here related to fungi as input into alternative protein products.
Yeah, so it's an important topic, not only for allergens, but also for potential toxins, right? So some of these species that I talked about today, Aspergillus niger, well, that's all the plant pathologists in the room will know that one well as a production of aflatoxin, which is a big pest actually for crops. And luckily, these types of toxins, these are part of the screening and selection criteria early on when you're looking at food product applications of fungi. I actually had a slide around strain selection that I didn't end up including in the presentation. Maybe now I'm kicking myself for taking out. But allergen production, also things like you might be aware for corn a QUORN mycoprotein. They have a step in the production process for getting rid of high levels of RNA by heat treatment of their things to avoid some health impacts in that end. As far as allergens are concerned, it's a pretty strain-specific thing that allergens and toxins. It's a strain-specific thing that will go along with the trials, as people are starting to eat that, but you can kind of get around some of that by being very selective in your, selection from the very beginning, working with organisms that, for instance, in the United States have FDA recognition as a generally recognized as safe organism.
Yeah, absolutely. We have a few big themes to touch on, and I'm just scanning through these 37 questions in our next chunk of time together. And I think we'll move next into discussing the kind of environmental impacts of mycoprotein production and how it might compare to our other production platforms. And then some questions regarding cost and scale and supply chain and ensuring, cohesion of end product when the inputs can be so variable and byproducts can be so variable.
Oh, yeah.
But maybe starting with the environmental footprint question. Do you know how the environmental impact, whether we're considering emissions or water use and land and fertilizers, et cetera, compares when it comes to the production of feedstock for mycoprotein production versus maybe the production of plant-based proteins? I'm certain both are significantly significantly more sustainable than conventional animal agriculture along those various dimensions, but curious if you know of any studies or figures that compare fermentation and plant-based production.
Well, I love this question because I get to plug my the second paper that I introduced on that one of the first slides there from food bioengineering around cell cultivated foods. And in that paper, there's a table that we put together around that very question, bringing together all the different life cycle assessments that have been done around in vitro animal cell, so cultivated meat production, fungi-based mycoprotein production, microalgae based, as well as including traditional animal as well as plant-based protein life cycle assessment. So that is an open access article, as we said, and I really direct you. It's like it might be the last table in that paper that will pretty directly answer that question. And, the broad strokes of this, in my opinion, are that we still have a long way. One of the things about doing life cycle assessments is that we try, and it's a standardized approach to do life cycle assessments for the very reason that it's hard to compare across different boundaries of different assessments, right? And I would say that in the protein space, it's not immune to that problem. So looking at a table like the one that you're going to find in my paper ends up having a lot of, I have to say, you have to look at those in the context of how the study was performed. And it's very difficult to compare them directly to each other. One, because of how the inventories are created to really quantify impacts like water use, CO2 emissions, and so forth. The scale at which, when you're comparing mycoprotein production, which is, compared to animal agriculture, is a tiny baby industry, even, even worse case for the animal or cultivated meat application, where, it's not at scale at all. And trying to project out results of what it's going to look like is quite challenging. So for that reason, you'll see a lot of variation in those results, and sensitivity and uncertainty analysis that are kind of critical parts of each one of those studies. But yes, you're probably right. The total taken as a whole environmental impacts at the moment of things like mycoprotein production are definitely much more favorable than things like beef production. That's kind of one of the highest environmentally speaking, emitting high amounts of carbon, using a lot of water. That's one of those that is an easier bar. So otherwise, in that paper, I think when you compare things like mycoprotein compared to something like chicken or poultry production for meat production, that's where the differences presently are less severe. But again, a lot of that could come from further scaling in the mycoprotein area that will, lead to the ability to, recycle more of the nutrients and energy from that sector, and achieve even better performance moving forward. So, it's a really hard question to answer like one fell swoop, but definitely if you have any questions from the table that's in that paper or anything like that, I'd be happy to talk with you further about it. It's really an important and interesting topic.
Yeah, thank you so much, Tyler, for adding so much nuance to what is a very big question. My colleague Renee dropped the link to the paper Tyler's been referencing in the chat for those of you who are interested in taking a look at the figures in that table. And my other colleague, Pri, also dropped in a really wonderful life cycle analysis assessment done by DIL, comparing corn to other forms of meat production. So that could be useful to those of you who are interested in better understanding the environmental impacts of these technologies as well. Staying on this theme, and then in the interest of time, I'll pivot us onto the next theme. You spoke on how resource utilization efficiency is lower for traditional plant agriculture versus bioprocesses when growing plants or microbes, respectively. So Pri's wondering if you can talk a little bit more about downstream processing of microbes to food ingredients and how this compares to maybe the process of crop fractionation. Which would you say is more efficient from a cost and environmental perspective? And do feel free to punt people to your paper if that's the best way to answer this.
Oh yeah, it's another really super question. So to start my answer to that question, the first thing that I'll kind of do is say, in many ways, so my idea that the flow chart that you saw, or whatever, the process diagram at the beginning, and this concept of achieving more resource efficiency in these types of bioprocessing systems, that's kind of our goal at all times, right? And by no means am I saying that we have reached that. Or that it's within grasp in the next 5 years or anything like that. These are the goals that things like my research are trying to achieve. And another interesting and important aspect to that question is, are you comparing apples to apples with two products? So when we are looking at mycoprotein and creating a burger type of product compared to a traditional animal type of burger. It's a pretty direct comparison that we can make between those two, and talk about the resource use efficiency. When you start bringing in new types of foods, and in some cases, I had this where we're exploring about the Boba or myco-Boba, aspect with fungi. like, I don't know. I mean, I don't have all the answers for the types of products. I'm really interested in like what types of totally new foods are going to be made that we don't really have a good direct comparison to historically. And that's a whole other kind of can of worms answer to that question. But going back to some of the things that I talked about in the presentation here, in ways that the bioprocess area can be made to be more efficient or to achieve good performance, is by achieving circularity, one with nutrients, the other with water, and the other with energy. That's something that I think should go into the design of each one of these. There's a lot of money in this space, and bringing the topic of how nutrients and energy and water are going to be used in these processes, and really circular ways being built into the design of that process, I think will get us a long way from there. And another thing is to that microalgae part that I talked about. So microalgae are microscopic plants, and you might be inclined to compare them in many ways compositionally to like a more traditional plant, right? And one of the problems that has historically come up with downstream processing, or even just getting to a microalgae paste, that I alluded to kind of briefly in this presentation, is that process takes a lot of energy. As you're going from, you're comparing like how we harvest a soybean to how we harvest microalgae. There's a lot of energy that has to go into the centrifuge type of process to harvest really microscopic cells. And that's a place where this immobilization using a scaffolding with a living fungal scaffold is kind of solving two problems at once. One, we get the algae easily taken out of the solution. We're just shaking this around. It's pretty easily. It's kind of magical when you see it happen. And then you're also creating like a beneficial component to this food product in the end. So I think finding more and more of those ways where you can eliminate some of the energy intensity that come into the processing, the harvesting, the other aspects of bioprocessing in general, that's going to really play a large role in these life cycle assessments and resource use efficiency moving forward. And I don't, we're not completely there yet, but I think we can get there.
Thanks, Tyler. Moving us along to supply chain constraints. Rumor has it there's a supply chain constraint right now for citric acid. So could you maybe speak to what kinds of bottlenecks in fungi production could potentially lead to these types of constraints, even for highly developed ingredients like citric acid, and maybe any advice for overcoming these bottlenecks?
Very interesting. Okay, so I unfortunately don't know the exact reason for citric acid production and its current bottleneck in today's world. But I can answer that question more generally, maybe just we often have issues with nutrients, right? So I mentioned here about fungi. To grow them, we need sugar, and we need a cheap source of that sugar. And it's not just sugar. The other really critical component is nitrogen. It's needed in much lower amounts compared to the carbon in sugar, but you still need a lot of it compared to virtually any other component of the media there. So if I had to guess, I would say that's not the bottleneck probably is not coming from, the reactors aren't big enough, the fungi is not productive enough, or something like that. They might be running into struggles in getting cheap sources of essentially fertilizer. We've seen fertilizer prices going crazy for traditional food products this past year as well. In some cases, farmers are paying dairy farmers for manure because they just simply can't get synthetic fertilizer, nitrogen sources for their crops. It could be that similar problems in the supply chain have arisen around sugar production or other components of that media. And that's something that we're always contending with. And, I think goes back to what was mentioned earlier about, using byproducts. And will that solve everything for us? Well, it makes things a lot, in many ways, it can make them much more complicated. We want to utilize the byproducts, but then you have this problem where it's like, well, it's more sustainable for us to use that. But then in order to make this a functioning business, we have to have a ready supply of this. We become dependent on this waste product to be made in large quantities. As we want to grow this industry, we need more and more and more. And, I think, so I've worked in the nutrient kind of this industry for a while. And I'll never forget one time when I was giving a tour of the anaerobic digester that's on the UC Davis campus. So I was a grad student there. And I was giving a tour to an elementary school students about, hey, you can take food waste and make it into energy and everything. Super cool thing. And then, of course, when you're distilling this idea down to that audience, I was amazed to be getting these questions. Well, Tyler, I thought that you said that waste was bad, but it sounds like I should just take my pizza instead of eating it, throw it in the trash, and it comes here to be made into energy. And so that's an example of like a perverse incentive that you don't want to end up creating, or an unforeseen kind of consequence of these circularity concepts. And I think that plays well into it's a cost thing, but also it can play a lot into the supply chain availability things when these come to fruition. So I don't know the exact answer to the citric acid question, but it's my 2 cents there.
That was so helpful. I wish I were getting those kinds of lectures as an elementary school student. The crop of geniuses that you helped seed in the world. But yeah, that actually touched on, I think it's fascinating. It's so true that there can be perverse incentives created in things that have kind of a halo around them. Like, our reverence for a circular economy. It's true that we don't want to be creating a reliance that this sector has on other sectors that might one day become obsolete, or make it so that they don't have incentives to become a lot more sustainable in their own production processes. So that's a really great set of flags, and appreciate those considerations. Honing in on the question of cost, which you've touched on a little bit already, you did mention that sugar is pretty much the biggest cost driver when it comes to production. And because sugar is expensive, I'm wondering if there are other solutions, low-cost substrates that you might recommend, if we're not looking at byproducts, what are we then looking at in terms of solutions for lower cost drivers as inputs into feedstocks?
Oh, I wish I had the answer. Yeah, it's so tricky to take byproducts out of that conversation. Then I think you have to start thinking a lot more creatively in ways that we're just not there yet, at least in how we handle microorganisms. And we're kind of very scared of bringing multiple organisms together. But, maybe my 2 cents to that is look into, so outside of byproducts, look into the biology of these systems a bit more creatively. The example that I gave today about the co-culturing of the algae and the fungi is maybe a good example of that, where, of course, when lichen are growing on the backs of rocks, they don't need to have the sugarcane industry to import a bunch of sugar for them, right? They have a relationship, the microalgae, sunlight, CO2, makes sugar, feeds itself, feeds the fungi produces CO2, feeds the microalgae. You have this happy family um, kind of situation. So bringing those two things together a bit more in depth, or being able to have one facility, for instance, the mycoprotein production facility making fungi, also having a site right there for producing and extracting sugars, for instance, from their microalgae on site. That might be a way where you can make use of a lot of the same infrastructure that's needed for those two different types of organisms, but they are kind of completely different, but they share a lot of commonalities. You need tanks and pumps and these types of things. They're similar enough that you might be able to kind of co-locate them and solve some of your problems with cheap CO2. Ultimately, the sugar that we're getting here is coming from CO2. And the ability to directly capture that on site might be one of the solutions there. But man, it's a good question.
Lots of heavy hitters coming your way today.
Yeah.
I think this next suite of questions really directed at the co-culturing of algae and fungi, which everyone was like, this is so fascinating. It's such a cool technology. But folks are wondering, for starters, how do you choose which species to cultivate? Are you selecting fungal and algal species that lichenize in nature? Is there something else you're looking for?
Great question. Yeah. So for the ones that I presented here, for instance, with that red, it's not technically, biologists will get mad at me if I call it a red algae, but that one that was red looking, Hemitococcus pluvialis, that's an example. I honestly don't know if that has a historical relationship or an evolutionary relationship with fungi in lichens in nature. Right off the bat. But the reason that was selected was because of its interesting, characteristics to bring good components to the final product. So the color there, the antioxidant property, as well as the other parts of that specific microalgae were reasons for selecting that. I think moving forward, there could be a huge amount of work that really brings us engineers and technologists kind of more in line with this great body of work that the biologists have been doing for years and years around lichens. And, lichens are not as simple as I made them sound either, in that there's usually two completely different classes of fungi that themselves are living together with at least one other species of microalgae there. So it's even more complex, where you're bringing in two pretty different types of fungi that live together in this happy family structure. And that's where the real interesting science is going to happen, I think, moving forward, is taking those relationships that have already been identified and bring them into some type of technological space for new applications. That's a really exciting space. I don't think it's been probed. Right now, we know that Aspergillus oryza is a generally recognized as safe organism. We know that Hemitococcus pluvialis is the same. It produces astaxanthin. Let's just throw them together, see what happens. And that's kind of where we are currently, at least for myself. Maybe some of my colleagues have better grasp.
Love that. Spirit of experimentation. I there are so many great questions left here, but I know we're quickly approaching the end of the seminar time. So maybe one last question for you on what it will take to really scale this fungi algae biotechnology. Can you speak to any of the pain points that scientists and innovators will need to solve for in order for this larger scale implementation? Maybe how bioreactor design influences the process, or yeah, just overall arching considerations, if possible in the next minute, and then we'll wrap things up with final thoughts.
Sure. Yeah. And thank you all so much for these great questions. It's been a lot of fun. All right. So it's always going to come down to cost. And it's in this space, one of the things that we have struggled with in the fungi and algae space is, really around finding, there's a lot from the research standpoint that needs to be done to, some of the things that I showed in the presentation to just, completely start from the bottom and how to put these two things together, model it, all of those things, be able to predict what we're going to get, right? But then in developing a food product for this and finding the right way to process these downstream and make them end up in the right product is a whole other very important process there. And there's a food aspect of that, but also there are other applications of that kind of technology that you might not consider. So the truth of how I got into growing fungi and algae together was I was trying to treat wastewater. So, or natural waters that had been eutrophied by microalgae, right? You go out to the lakes and they have this gross green scum everywhere. I can dip a tea bag of fungi in there and clean it all up, right? It sounds really good on paper, but how do you make money from that? That's where the rubber hits the road for the business development side of things. And I think that generally speaking, the technology around bioreactor design and the ability to scale up these types of technologies is at sufficient maturity to allow for that. We need more product and development on the back end to find the right application for these things. Yeah, so that'll be my 2 cents there.
Yeah, thank you so much, Tyler. There's a tremendous amount of gratitude coming from our community for everything you've shared today, and just your time, your generosity. Really appreciate everything you've done in service of advancing a more sustainable food system, and appreciate you highlighting the questions that still remain for other scientists to kind of tackle in this collective quest. So with that, it brings us to the end of our seminar. Thank you so much to everyone for joining us. Thank you so much to Dr. Tyler Barzee for this phenomenal seminar. The recording will be up on YouTube and sent out after the seminar. So for those of you who are interested in joining, continuing the discussion, feel free to email Tyler and or join the networking session that's starting now at the Meetaway link that Renee has just dropped there in the chat. We're looking forward to continuing this discourse with all of you. So thanks so much, everyone. Take care. Bye.